Hello friends, today we shall discuss about chapter number 5. In this clip, we shall concentrate on monohybrid cross or one gene inheritance. In order to understand the principles of inheritance, one should know some basic definitions. Let's see few of those definitions. First one is contrasting traits or characters. These are pairs of alternate traits. So every character has a pair of alternate traits. For example, height is a character which has two contrasting traits, that is tall and dwarf. Now, these contrasting characters are controlled by a pair of factors. Now, Mendel used the word factors. So, these factors are now known as alleles. Okay. A pair of factors are responsible for a particular character. Always remember that any character is controlled by a pair of alleles or a pair of factors. For example, for height, there are two factors, capital T and small t. Now, this capital T or dominant allele controls the tallness, whereas the small t or recessive allele controls the dwarf character. Phenotype. Phenotype is nothing but the external appearance of a particular character. Say, for example, height has two alternate phenotypes or contrasting characters. One is tall and the other is dwarf. Now, genotype is nothing but the genetic constitution responsible for the phenotype. Okay, it is nothing but a pair of factors or alleles that control a particular phenotype. Say, for example, this tallness. Okay, the tall character is controlled by two types of genotypes. One is homozygous dominant, and other one is heterozygous. Whereas this dwarfness is controlled by two recessive alleles, small t and small t. Now, homozygotes. Let's see few more definitions. When the alleles for a particular character are similar, we use the word homozygous. Here you can see alleles for a allele pair for a particular character, they are similar. Here also they are similar. Then we use the word homozygous. It may be homozygous dominant or homozygous recessive. If allele pair contains both the dominant alleles, then we use the word homozygous dominant. Or if the allele pair contains both the recessive characters, then we use the word homozygous recessive. When the alleles for a particular character are dissimilar, here you can see one is dominant allele, another is recessive allele, we use the word heterozygous. So when the alleles are similar, homozygous, when the alleles are dissimilar, then we use the word heterozygous. Now the question arises, where are alleles located? Well, alleles are located on specific loci. Here you can see there are specific points on the homologous chromosomes where you will find the alleles. Okay. So alleles are located on specific loci. Here you can see alleles are located on specific loci of the homologous chromosomes. As you already know that in diploid organisms, chromosomes occur in pairs. Each of these pairs is called homologous. So humans, in human females, there are 23 pairs of homologous chromosomes, whereas in human males, there are 22 pairs of homologous chromosomes. Okay. Now here, this is a pair of chromosomes. We have shown a pair of chromosomes here. Three allele pairs are shown here. Here you can see T, A, B. Okay. So all these allele pairs control a particular character. Here one has to remember that each allele pair is located exactly on the same location. Say for example, allele pair for a particular character. Okay, height. They are located on the same positions on the chromosome. Now these positions or locations, they are called as loci or locus. During gamete formation, these homologous chromosomes separate. So during gamete formation, what happens? One gamete receives this homologous chromosome, other gamete receives this homologous chromosome. When this happens, even the alleles which are located on the homologous chromosomes also separate, so that each gamete receives only one copy of the allele pair. So this is one allele pair. Since this homologous chromosome containing this allele goes to one gamete, then this allele will go to the other gamete. So alleles, they are separated during gamete formation. Now, Mendel studied 7 pairs of contrasting characters in garden pea plant. Now they are listed here. These characters are flower color, seed shape, pod color. Pod means fruit of the leguminous plants. Okay, and pod shape, flower position, and plant height. So these are the 7 pairs of contrasting characters. So out of these, few will be dominant, few will be recessive. So the dominant phenotypes are shown here, okay, whereas the recessive genotypes, they are shown below. Okay, for example, flower color. Okay, purple flower color is dominant over white flower color. Yellow seed is dominant over green seed. Okay, round seeds are dominant over wrinkled seeds. Green pods are dominant over yellow pods. Inflated pods are dominated. Okay, they are dominant over constricted pods. Axial flowers. Okay, they are dominant over the terminal flowers, and tall character is dominant over the dwarf character. Mendel selected, so here we are shown the representation of a monohybrid cross. Mendel selected a purely breeding tall plant with, okay, he crossed a purely breeding tall plant with purely breeding dwarf plant. Okay, since the two plants differed only in one pair of contrasting characters, that is tall and dwarf. So we have considered only height here. Okay, only one character. So these plants, rest of characters are same in these plants, but only difference is this plant is tall, this plant is dwarf. That is why it is called monohybrid cross, because we have selected the plants which differ only in one character. It is also called inheritance of one gene, so which is represented here. Now, tall plant produces gametes containing dominant allele. Okay, so it is purely breeding tall plant. It produces gametes containing dominant allele. So all the gametes contain dominant allele here, and dwarf plant produces gametes containing recessive allele. Now, when these gametes fuse, okay, so gametes are shown in the Punnett square here. Okay, along one axis. Okay, here as you can see the gametes of females or males is shown, and similarly the gametes produced by males or females. Okay, generally it is the female gametes and this is the male gametes. Then when we combine them, we will get the possible genotypes in Punnett square. Okay. When when these gametes fuse, okay, F1 generation is produced. He observed that all the plants in F1 generation are tall. So no plant in the F1 generation was dwarf. All plants were tall. Okay. Then he self-crossed. Okay, he self-crossed F1 plants. He self F1 plants. Okay, so F1 plants were tall. So he took two F1 plants and crossed them. Okay. In F2 generation, okay, here you can see F2 generation. In F2 generation, he observed that the ratio of plants, okay, ratio of tall plants, here you can see the ratio of tall plants. So all these are tall plants. This is dwarf plant. The ratio of tall plants to the dwarf plants was 3:1. Means there were three tall plants and one dwarf plant. That is 3/4 of the plants were tall. Okay, here you can see this is tall, this is tall, this is tall, and only 1/4 of the plants was dwarf. Okay, so the phenotypic ratio was 3:1, means 3 tall, 1 dwarf. So here you can see in F1 generation this dwarf character was masked, but in F2 character this dwarf character again recovered. Now, based on his observations in monohybrid cross, Mendel formulated two laws, namely law of dominance and law of segregation. Law of dominance states that In heterozygous condition, one of the allele expresses itself and suppresses the other allele. So one of the alleles, okay, which now we call as dominant allele, expresses itself and suppresses the other allele. Okay. Now this law was based on the fact that in F1 generation, here you can see in F1 generation, even though dwarf allele was present, here you can see dwarf allele was present. Okay, in all the genotype dwarf allele was present, but this dwarf allele did not express itself. Instead, only the tall allele expressed. So he concluded that one of the allele suppressed the effect of other allele. Okay, now we know that this dominant allele has suppressed the effect of the recessive allele. Okay, so he concluded that one of the alleles suppressed the effect of the other allele. This allele he called as dominant allele. Now here capital T is dominant allele. Okay. And the other which was suppressed, now here the small t or recessive allele, the other which was suppressed in the presence of dominant allele was called recessive allele. Okay. Now the second law or the law of segregation, it states that each gamete receives only one copy of the alleles. That is, the alleles do not blend. Okay, it means gametes are pure. So we'll see once again. Each gamete receives only one copy of the alleles. Now concentrate here. Take, say for example this. So this is the allele pair which is located on homologous chromosomes. Now we know that these alleles, okay, they get separated during gamete formation in such a way that each gamete contains only one copy of the allele. Only one copy of the allele. Here you can see. So for example here the dominant allele goes to one gamete, whereas the recessive allele goes to another gamete. Like this, each gamete contains only one copy of the allele. That is, the alleles will not mix in a single gamete. They get separated. That is why we use the word gam. We say that gametes are pure. That is why law of segregation is also called law of purity of gametes. Okay, that is because these alleles do not mix, do not blend. They are separated during the gamete formation. Okay. Now, test cross. Test cross is a cross between F1 or F2 plants showing dominant phenotype. Okay, with the double recessive parent. So here F1 or F2 plant with unknown genotype is crossed with double recessive parent. It is done in order to determine the genotype of plant showing dominant phenotype. Okay, so here you can see that homozygous dominant plant also it is tall. Heterozygous plants are also tall. So we don't know if there is a tall plant, we don't know whether it is homozygous dominant, its genotype we don't know, okay, or it is heterozygous. So to determine whether the F2 or F1 plant is heterozygous, F1 plant showing dominant phenotype, whether it is heterozygous or homozygous, we conduct test cross. Okay, in this example that is shown here, color of the flower is taken into consideration. In garden P, the flower color is taken into consideration. So we know very well that in garden P, violet color is dominant over white flower. Okay, so violet is dominant over white. The allele for violet color is represented by capital W. Allele for violet flower is represented by capital W, whereas capital W or uppercase W. Okay. Whereas the lowercase w, okay, it represents the recessive allele. It represents recessive allele. In both homozygous dominant and heterozygous condition, violet color flowers are produced. Here you can see. So this violet color flower, okay, it can be produced in under both genotypes. Say let it be homozygous dominant or heterozygous. So in both cases, okay, in both the genotypes, purple color flower is produced. But we don't know whether it is homozygous dominant or recessive. but we don't know whether it is homozygous dominant or heterozygous. Sorry, whether it is homozygous dominant or heterozygous. That we don't know. To test this, the plant showing dominant phenotype, so that is purple color flower here, is crossed with double recessive parent. So double recessive parent means homozygous recessive parent, which shows the recessive phenotype, that is white color flower. If it is homozygous, then all plants will produce violet flowers. See, if it is homozygous dominant, then all the flowers in F1 generation of test cross will be violet. If this occurs, then we can say that it is having a homozygous dominant genotype. Okay. If it is heterozygous, here you can see, if it is heterozygous, when it is crossed with the double recessive parent, then half of the flowers are violet and half of the flowers are white. So that is nothing but violet flowers and white flowers are produced in the ratio 1:1. So if the ratio is 1:1, then it is heterozygous. If all flowers are violet, then it is dominant phenotype. Now let's solve few questions related to monohybrid cross and test cross. Question number 1. Which of the statement is correct? Each back cross is a test cross. Each test cross is a back cross. Crossing F2 with F1 is test cross. Crossing F2 with P1 is called test cross. Now remember, each back cross cannot be a test cross, because in test cross the F1 or F2 hybrid is crossed only with double recessive parent. But in back cross, F1 and F2 parents is F2 hybrid is crossed with either double recessive parent or homozygous dominant parent. But in test cross, okay, test cross is also back cross. Okay, test cross is also back cross. So this is the correct answer. Now remaining options, let's see. Crossing F2 with F1 is a test cross. No. Crossing F2 or F1 with double recessive parent is nothing but test cross. Crossing F2 with P1 is called test cross. No. So crossing F2 with P1, okay. P1 means here both double recessive parents as well as no, they are considered P1, right? Yes. So this is not a test cross. Now this will be a back cross. Remember, when F1 or F2 hybrid, it is crossed with the double recessive parent, then it is called test cross. So here the answer is second one. Each test cross is a back cross. Now, in garden P, round shape of the seeds is dominant over wrinkled shape. A pea plant heterozygous for round shape of seed is selfed and 1600 seeds are produced. During the cross, okay, 1600 seeds are produced during the cross, which are subsequently germinated. How many seedlings would have the parental phenotype? Now we know here it is given that in pea plants, round shape of the seed is dominant over wrinkled one. So here uppercase letter capital R represents the dominant allele, and this lowercase letter small r, okay, it represents the allele for wrinkled seeds. As mentioned in the question, two heterozygous plants, okay, two heterozygous plants, here you can see both are heterozygous, which are showing the dominant phenotype here. Two heterozygous plants for round shaped seeds are self. In the next generation, we will get the round and wrinkled shaped seeds in the ratio 3:1, because it is a typical monohybrid cross. Okay, it means 3/4 of the plants, you can see 3/4 of the plants show round seeds, which is nothing but parental phenotype. Okay, and remaining 1/4 are wrinkled, which do not show parental phenotype. So these are the parents. Parental phenotype means they should show round seeds. So 3 out of 4 show round seeds. Okay, so he in the options, okay, total number of seeds they are given is 1600. So 1600 multiplied by 3 is to 4, because 3 is to 4, sorry, 3 by 4. So 3 out of 4, okay, are showing round seeds, which is nothing but parental phenotype. So you multiply 3 by 4 into multiply 3 by 4 with 1600, you will get 1200. So 1200 is the correct answer here. Next, the genotype of a plant showing dominant phenotype can be determined by. So this we already discussed is nothing but test cross. So here we don't the unknown genotype, okay, which is showing the dominant phenotype, it can be determined in the test cross. Okay. Mendel's principle of segregation means that the gamete cells always receive one pair of alleles, one quarter of genes, one of the paired alleles, any pair of alleles. So here the answer is we already know. It is also called Mendel's law of purity of gametes. So here what happens? Okay, in the principle of segregation states that each during gamete formation, the alleles get separated in such a way that each gamete receives only one copy of the allele, that is one of the paired alleles. Okay, so not complete gametes will not receive the complete pair of alleles, okay, complete one pair of alleles, but out of this pair one is received by the each gamete.